Okay, everyone, so welcome to the third installment of Flipping the Classroom. We are going to deal with the second half of Chapter 8.3, which is the Calvin cycle. Okay, just as a reminder, we've already talked about the light reactions that occurs inside of the thylakoid. Now we're moving out of the thylakoids and into the stroma. We're still inside of the chloroplast, okay, except we're in a different part of the chloroplast called the stroma. Again, takes place in the stroma. Your Calvin cycle is going to be powered by the energy from ATP and NADPH, those molecules that we made earlier in the light reactions. It's going to make high energy sugars such as glucose or sucrose or starch, okay, a lot of these sugars, and it's for long-term storage of all of this energy from the light that the plant is getting. Your Calvin cycle does not require light, however. Remember, this is the light-independent reactions. The only part of photosynthesis that requires light are those light reactions that we spoke about earlier in the thylakoids. Your Calvin cycle can occur in perfect darkness. It does not require light. It does, however, need carbon dioxide. Okay, to be exact, it needs 6 molecules of carbon dioxide. It also needs 18 molecules of ATP from the light reactions, and it's going to need 12 NADPH also from the light reactions. And all of these molecules are going to be used to make only one glucose molecule. Okay, so from 6 carbons, 18 ATPs, and 12 NADPH, you're going to get one molecule of glucose. The important enzyme to know is Rubisco, and it's going to just catalyze basically the very first step. Remember that enzymes are proteins, and if you're talking about an enzyme, it speeds up the rate of a reaction. Okay, this is all Chapter 2 review. So your Rubisco is a very important enzyme because it starts the Calvin cycle. All right, let's take a look at this diagram real quick before we get into the different steps. Again, the first part of photosynthesis inside your chloroplast, this big bean-looking thing is your chloroplast. Okay, the very first reaction are the light-dependent reactions. These are the ones that need the light from the sun. It also uses water in order to make oxygen, which is released as waste. That's why it's going out of the chloroplast. It makes ATP from that hydrogen ion gradient, and it makes NADPH. That ATP and NADPH is going to leave the thylakoids and enter into the stroma where it's going to take place, it's going to play a part in the Calvin cycle. In your Calvin cycle, you're going to need carbon dioxide. You're going to need that ATP and NADPH in order to make your high energy sugars, your glucose, your sucrose, your starch, your blah, 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 blah. A bunch of different types of sugars in order to store all of this energy. Anything that's left over after it's been used, right? So your NADPH gets turned into NADP plus once it's used. Your ATP turns into ADP once it's used, and any phosphates that are left over are going to go right back into the thylakoids so that the light reactions can recreate those molecules to be used. Okay, this is why these two cycles are dependent upon each other. They kind of are one big giant loop-de-loop, reusing and using the materials that are available to them. Okay. So the first step of the Calvin cycle is very, very important. It is when 3 molecules of carbon dioxide, remember CO2 is carbon dioxide, are taken from the atmosphere, in other words, taken in from the air, and it's going to combine with a 5-carbon compound called ribulose bisphosphate. It's also known as RUBP because that name is ridiculously long, so we have to shorten it. Okay, so 3 molecules of carbon dioxide will each combine with 1 molecule of RUBP. So you're going to need 3 molecules of RUBP as well at this point. Okay, so each carbon dioxide combines with 1 RUBP molecule. It's catalyzed by Rubisco. Okay, again, it is catalyzed by Rubisco. Hint, catalyzed by Rubisco. Okay, so your Rubisco enzyme is going to make this go a lot faster. That's what a catalyst does. That's what an enzyme does. This step is also known as carbon fixation because you're taking the carbons from carbon dioxide in the air and you're going to fix it onto RUBP. Okay, I understand that fixing means to make something better that may be broken, but in this case it means to put something onto something else, right? To attach something. So carbon fixation, meaning to take carbon from the carbon dioxide and attaching it to the RUBP molecule. What you get in the end are three molecules of, three of these molecules that have 6 carbons each. Okay, so the result is six carbon molecules. I'm going to be throwing a lot of numbers at you guys. Okay, so this doesn't mean that there are 6 little molecules of carbon. That means that there are 3 big molecules each made of 6 carbons. Okay, so let's review this again. Carbon dioxide comes from the air, 3 of them. They combine with RUBP, 3 of them. This is called carbon fixation and catalyzed by Rubisco. And what results from this are 3 6-carbon molecules. Okay, again, 3 large molecules made of 6 carbons each. The second step is that these molecules, these 6-carbon molecules, are going to break themselves in half because they are very, very unstable. They don't stay a 6-carbon molecule very long. Okay, this forms 6 molecules that are 3 carbons long, and the name is 3-phosphoglycerate, shortened to 3-PGA. Okay, now if I ever quiz you on this, you never have to write the big old long name. You just have to recognize what 3-PGA or RUBP are. Okay, I'm not going to have you guys memorize all of these names. Okay, so again, these 6 carbon molecules split in half to form 3 carbon molecules called 3-PGA. Here's a diagram to show you what's going on. I know it's a big old mess and it's confusing, but we'll go through it step by step. 3 carbon dioxide molecules enter into the Calvin cycle. It's going to combine with RUBP, catalyzed by that enzyme Rubisco. As it continues through the Calvin cycle and is combined, it forms a 6-carbon intermediate, a 6-carbon molecule that breaks in half to form 3-phosphoglycerate, or 3-PGA. Okay, so those are the first 2 steps. Carbon dioxide combines with RUBP, molecule breaks in half, forms 3-phosphoglycerate. Once the 3-phosphoglycerate are formed, the 3-PGAs are formed, ATP is then going to lend some of that energy that it has to continue the reactions. Okay, so ATP is going to add a phosphate to each of the 3-PGAs, and what you form is what's called 1,3-bisphosphoglycerate, BPGA. Right? Thank goodness for abbreviations, right? So ATP adds a phosphate to each of those 3-PGAs to make BPGA. The ADP then moves back to the thylakoids. Remember, once you use ATP, it becomes ADP, and it moves back into the thylakoids to be reused. The next step, you're going to use NADPH. You're going to take its electrons. It's going to be donated into BPGA. Sorry, that's supposed to be BPGA. And it turns it into a molecule called G3P, otherwise known as glyceraldehyde 3-phosphate. The NADP plus that forms from this goes back into the thylakoids. So every 3 molecules of carbon dioxide that has entered into the Calvin cycle is going to give you 6 molecules of G3P. One of those molecules goes on to make glucose. The other 5 regenerate RUBP because you need this molecule of ribulose bisphosphate in order to continue the Calvin cycle. If you don't regenerate RUBP, the Calvin cycle is going to stop. Okay, you're going to need 2 G3Ps in order to make 1 glucose molecule. Okay, because remember, glucose is a 6-carbon molecule. G3P has 3 carbons, so the Calvin cycle has to turn 2 times to make 1 glucose molecule. Again, a lot of numbers. I'm going to have a little animation diagram thing at the end. Hopefully, it'll help you guys visualize this better. So don't freak out on me just yet. Okay, here again is the diagram. We are here last time at 3-phosphoglycerate. ATP comes in, donates its phosphates, become ADP. That gets reused. It forms 1,3-bisphosphoglycerate, or BPGA. NADPH donates some of its electrons 2-BPGA turns it into G3P. One of the G3Ps go on to make sugar and other such compounds. The other 5 goes on to remake RUBP. Okay, and that process also requires 3 more ATPs to be added. Right? Alright, so let us review this one more time now with more pictures. Yay, pictures. Okay, so hopefully this will help you guys kind of straighten it out in your heads. If not, please, 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 for the love of all that is holy, come ask me questions, okay? So the first step, again, carbon dioxide combines with RUBP. So here are the CO2 molecules represented in blue, coming from the air. And these purple and orange molecules are RUBP, the ribulose bisphosphate. Okay, it has 2 orange phosphate groups, 1 on each end. Again, this all happens in the stroma. The thylakoids are over here, so we are not in the thylakoids, we are now in the stroma. Okay, so this is what happens. Your carbon dioxide molecule combines with the RUBP. It forms this unstable 6-carbon molecule. Okay, so we're going to count 1, 2, 3, 4, 5, 6 carbons. Now, because that 6-carbon molecule is so unstable, it's going to shake, it's going to vibrate, and then it's going to break itself in half. And this is step 2. Okay, step 2, your 3-PGA, okay, 3-phosphoglycerate, is going to be formed, right? These are 3 carbon molecules. See, 1, 2, 3 carbons. 1, 2, 3 carbons. Okay, so basically, all we did was take this gigantomolecule and break it in half. Okay, so that each of these are 3 carbon sugar, or 3 carbons long. The third step, you get your ATP that comes from the thylakoid, from the light reactions. Okay, and it's going to add its phosphate onto the ends of these 3-PGA molecules. Okay, once it does that, it forms this 1,3-bisphosphoglycerate, and all the ADP molecules are going to return to the thylakoids to be reused for making more ATP. Okay, again, this is called BPGA, these molecules right here. They have 1, 2, 3 carbons still. NADPH is then going to come from the thylakoids with its little electrons. These little red dots represent electrons. Okay, and it's going to donate those electrons into your BPGA molecule. When it does that, okay, the electrons will pretty much pop off those extra phosphates, and those phosphates return to the thylakoids along with the NADP+, so that they can be reused. That phosphate gets added back onto ADP in there, okay, and it gets reused. When that happens, what you have are G3P molecules. 1, 2, 3 carbons, okay? That's step 4. Step 5, well, still in step 4, sorry. One of the G3P molecules is going to go on to make glucose. Okay. The other 5 G3P molecules will continue into the Calvin cycle in order to form or reform RUBP, and it needs ATP to help it with this. So 3 more ATP molecules come in from the thylakoids. They are going to donate their phosphates, okay, and it's going to make 1, 2, 3, 4, 5 carbon sugar, or 5 carbon molecules, RUBP. 3 of them, 1, 2, 3. Okay, if we go back and we count, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 carbons here. If you add on those extra phosphates from ATP, you pop some off, blah, 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 you're going to have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 carbons again. Okay, so all of that is being reformed. Hope that made sense. Alright, so a summary, really quickly, again, 2 turns of the Calvin cycle makes 1 glucose. You're going to need 6 carbon dioxide, 3 of which are required for each turn. Rubisco catalyzes carbon fixation. ATP and NADPH come from the light reaction, provides energy. Once they're used, they go right back to the light reactions to be remade. And after one turn of the carbon cycle, you get 6 molecules of G3P. One, again, helps to make half of a glucose molecule. That's why you need 2 of them. And the other 5 help to regenerate that RUBP molecule. All right. Again, questions, please ask.